All right, welcome back to Side Nerds Podcast, and this is going to be our podcast just for DNA, since we had to break this unit up into segments. But just before we go on, think back to when we started going over genetics. We said Mendel is the father of genetics, even though during his days when he was alive, he never got the credit that he gets now. But it's just I bring that up to establish a premise that to every concept in science, there's a building block that leads to what we understand today. For example, DNA. There are several building blocks that was put in place by several people over the years that have paved the way for us to to have the understanding that we have of DNA today. So just briefly, we're gonna go through some names quickly. I'm not big on names, but just briefly we'll go over through some names and see what foundation they've set in stones for DNA. All right, the first name we're going to talk about is Friedrich Griffith. Griffith, let me find a different color. Griffith actually, it was not until after World War One before DNA, you know, developments in DNA actually started to, hit the throttle. And Griffith actually started his work with his attempt to explain why pneumonia was killing people, and first of all, how it infected people. And if you know how something actually infects people, you might be able to find out how to deal with that problem. So Griffith specifically wanted to know about pneumonia. And so what he did, he looked at two strains of pneumonia that he tested on mice. And from the outside appearance, one of the strand was, kind of jagged edges. So, you know, he called it the rough strand. The smooth edge one, he called it smooth strand. He injected these two into into mice, and the rough strand, did not kill the mice, and the smooth strand did kill them. And so he realized there was a difference between the two, rough strand and smooth strand. And so, well, we said, the smooth strand is deadliest. Well, what he did, he heat killed or he heat treated the smooth strand and then injected the sample into mice. Those mice did not die because they never got pneumonia. So he proceeded to actually mix in the smooth strand with the rough strand. And when he did so, the mice actually did die. Um, because we know that, you know, rough strand by itself did not kill mice. Smooth strand heat treated or heat killed did not kill mice. But when you mix the two, well, mice die. How? Um, well, Griffith came with an explanation that the deadly part of the smooth strand must have been transferred over to the rough strand to make it, you know, deadly. So that transferring is actually called transformation. So as we talk about Griffith, Griffith is given the credit of actually the guy who pretty much discovered transformation, if you might say. Um, one strand of the bacteria had been permanently changed, by the other strand. You know, look at it this way: two siblings, all right, one very nice kid and one kind of a devious kid. Well, if the devious kid hangs out with the not so nice kid, um, over time they might be influenced by each other, but the devious kid might more so influence the other kid. If you start seeing the nice kid start doing things that are questionable behavior, you know that something changed. Something they got transformed, you know. Oh, look at the Transformer series. You know, a semi truck going down the highway and suddenly turn out to be a deadly killing robot, that is out to kill other robots. Transformation. So, you know, think Griffith, think transformation. So pretty much looking at all of this, we know that when Griffith actually did his work, uh, it was, you know, when Griffith actually did his work, we know that, you know, it was pneumonia that was in those strands. Those were pneumonia bacteria causing the mice to die. And probably what was in the pneumonia bacteria in a smooth strand that was making it deadly is it has to do with genes. We know that today. But unfortunately for Griffith, he did not know at the time that, you know, what was being transferred is genes. So this is one of the reasons why, you know, I urge you guys to always take good notes, is that every good scientist will have a good, you know, paper trail, because what happens is somebody else will come after you and they will be curious about what you did, and you want them to have a good start. You don't want them to start over from scratch. So the guy who came next in the picture is Oswald Avery. And Avery, oh, that's a bad circling. Avery actually repeated Griffith's work. Reason, he wanted to know what was being transferred, because Griffith knows something was transferred, but he didn't know exactly what was being transferred. So Avery's like, well, we just can't leave this here. You know, it begs for more experiments. So, you know, Avery actually decided to continue Griffith's work so he would know what was being transferred. All right. Basically, every experiment was more of a process of elimination. He made extract from, no, this is not milkshake. No, no, we can't eat this. He made extracts from the heat kills bacteria. And from there, he destroyed the proteins, the carbs, the lipids. All right. Well, what did these guys remind you of? These guys are actually called micromolecules, as we talked about them back in September. Ooh, there's only one September in the year. All right. When he destroyed the proteins, the carbs, the lipids, and he injected the extract into the mice still got pneumonia and died. So we know transformation still occurred. All right, because again, bacteria was still being transformed. So that leaves one explanation. The proteins, the lipid, the carbs, and the lipids, these macromolecules in an organism, pretty much in a, or these macromolecules do not really, they have nothing to really do directly with the killing of the mice or the transformation of a pneumonia bacteria. So he didn't just stop there. So what he proceeded to next was he destroyed the DNA in the extract. And one of our macromolecule, again, is what is, as we know, it's called nucleic acid. And so nucleic acid, an example of a nucleic acid is DNA. So he went in there and destroyed the DNA. And then he injected the mice. The mice did not get pneumonia, neither did it die, and therefore he concluded that transformation did not occur. Therefore, DNA must be the transforming molecule. Coming back to Griffith's work, DNA must have been what actually was being transferred. All right, but there's more evidence needed to actually directly explain how that happens, or how that happened. Sorry. Hershey and Chase were our two American scientists that did some of the most important experiments concerning DNA. Well, we're just gonna, you know, briefly go through these guys that work, because we don't have that much of, you know, time investment. Well, they studied viruses. All right, viruses are cool, like bacteria, but these guys specifically study how, you know, they manipulated viral DNA and bacterial DNA. They studied bacteriophages, and that is when a virus infects a bacteria. Now look at it this way: bacteria, you think you're a bad guy? I got a dude that's worse than you. Let's see how you do. Viruses do not have their own mechanism to keep themselves alive. They have DNA, and that's one of the things these guys found out with, viruses and bacteria. You know, they have their own genetic material. But bacteria had they have the mechanism to survive, because they are cells. They have you know, ways of making energy. They get ways of getting rid of waste. They get ways of taking in nutrients. Viruses don't really have all those, you know, mechanisms to survive, because if you can't take in nutrient, you can't get rid of waste. You can't make, you know, you can regulate your own process. Too bad. So viruses have this unique thing of actually that's how we get a virus infection because they come in and become a part of us and live off of us or whatever host they infect. So knowing that about viruses, these guys, decided to experiment with viral DNA and bacteria DNA or RNA to figure out what is it actually in a DNA or RNA. First, they figured out that bacteriophages have DNA or RNA as their core, and these are surrounded by protein coat. And the protein coat is actually a protective coating to keep anything from getting to their DNA or RNA. So if you're actually going to kill the virus or the bacteria, this is the first guy you're going to have to lay out their protective coat or the outer barrier or barriers in this case. So how does this happen with bacteriophagy? Well, imagine a bacteria DNA as a spaceship. I kind of like sci-fi stuff, you know, and all of a sudden the invader aircraft comes in and secretly land on top, not undetected, and they drill through the roofing material and then they send in a spy, and that spy goes in and blends in to be one of the home base soldier, and eventually they're going around and changing everybody's mind within the home base spaceship. Well, viruses do that. Viruses actually inject their genes inside of the inside of the host, and as the host make more of their own DNA, or the gene as they go through DNA replication, they make more of those viruses, and before you know it, you have more viral DNA. Sorry, you have more viral DNA than you actually expect, and over time they will take over. And when a virus make the bacteria or that host cell to explode, they release all that viruses or the viral DNAs out into the host organism. Well, Hershey and Chase said if they could figure out what was being injected into the cell, they would know what genes were made of. Are they made of DNA or are they made of proteins? Well, they grew viruses in the culture containing radioactive phosphorus and sulfur. Proteins contain no phosphorus, but DNA contains, also on the flip side contains no sulfur. So if sulfur was found in a dead cell, then the genes would be found in protein. But if phosphorus was found in a dead cell, then DNA would be, the guy. Because as we will soon talk about DNA, the backbone of DNA is actually, it has a phosphate group in it. All right. So that's one of the premises, one of the things that we learn from this. All right. Hershey and Chase mixed the viruses with the radioactive marker and the bacteria, and the virus, they let the virus inject the bacteria, then blah blah blah, they separated the virus and the bacteria, and blah blah blah. You know, the bacteria actually contain radioactive phosphorus, and like I said earlier, just seconds ago, if it was phosphorus, then it would be DNA because DNA has phosphate in it. All right, so Hershey and Chase conclusion was DNA contains genes and not protein. And we will learn soon, you know, in weeks to come that, that DNA makes protein. Everything we have inside of our body has to do with protein. Pretty much your immune system, how you function, everything you're going to do, you're going to need some form, shape, or size of a protein to function. And that instruction for making that protein or for making those proteins come from the DNA. So, yes, proteins does not make DNA, but DNA makes protein, and the information for making, proteins is contained in the genes, which is actually stored in the DNA. The DNA is a big storage box that have the program into how to make an organism or have an organism survive its environment. One of my favorite unsung heroes of history is Rosalind Franklin. Rosalind Franklin, just like every graduate student and postdoctoral students in the modern day research labs, we become modern day slaves, and I know some people don't like the word slaves or the euphemism of modern day slavery, but that's what you are. You do a lot of work and get little credit and little to no pay. Well, Rosalind Franklin actually worked for Watson and Crick, two two guys we're gonna know about soon. All right, she took an X-ray image she was the one that took the first X-ray image of DNA, and what she saw was that it showed an X-shaped looking thing, and which suggested to her that there must be a double strand. And if you look at the actual diffraction, you will see some little bit of a shadowy outlining, and it showed that there are nitrogen bases that were actually in the center of the two DNA strands. All right, so Rosalind Franklin actually discovered that the three-dimensional shape of DNA, showing that we have two strands with nitrogen bases in between of them, and you're like, what is nitrogen bases? Well, one step ahead, our friend Chargaff, he discovered the percentages. Well, nitrogen bases are the four ingredients in our DNA that make up our genetic codes. So what makes us different from other organisms and each other is it has to do with the arranging or the arrangement of those four nitrogen bases. And the four nitrogen bases are guanine, cytosine, adenine, and thymine. And so we abbreviate them as A, T, C, and G. A for adenine, T for thymine, C for cytosine, and G for guanine. Well, what this guy did actually was that he came up with this postulation or this rule that says A will always form a bond with T, and C will always link up with G in between those two ladders of our DNA, because DNA is more like this, and in between these, all right, that's what he's talking about. If A is over here, then there will be a T over here. If C is over there, there will be a G over here, and so forth and so forth. Imagine those are in between the lines. I'm just putting them outside the line just because of space. What first? Let's take one step back. Actually, well, how did Chargaff find out his base pairing? Well, he looked at the concentration of adenine to thymine. They are very close to being the same percentage wise and in concentration. Sorry, in concentration wise. And when it comes to the concentration of cytosine or guanine, they're strikingly close to resemblance of each other. And so he came to the conclusion that if these two have close similarity, then they must be, they must form similar bond. And if these two has close similarity, they must have similar bonds. This brings us to Watson and Crick, the two guys that when you hear DNA and their names come singing out loud, well, they didn't start all of this. Going back to our girl Rosalind Franklin, they used her X-ray image to actually build the first three-dimensional model of DNA, as some of you would do for your project. All right, so the model was a double helix. That means two strands twisted around each other. They look like a ladder because if you unfold this, you will have something parallel to each other. But we know that DNA runs in a opposite direction of each other, but it's a ladder. The hydrogen bonds are in the middle here, holding your nitrogen bases or your A, T, C's, and G together. We use a hydrogen bond because it's as we will talk about a little bit soon in a few minutes, it's a much weaker bond, and we don't want something so strong. All right. And this brings us to the components and structure of DNA. What does DNA stand for? Well, DNA stands for deoxy, ribose, nucleic. Sorry, my handwriting is worse than yours today. Nucleic acids. All right. DNA. So when you hear DNA, it's an acronym, or sorry, it's an abbreviation of something much more complex. What makes up the components of a DNA? Well, a DNA, before we get into that, a DNA must do three things. Always. DNA must carry genetic information from one generation to the next. We found that out with Mendelian genetics with inheritance, and this is where you pass on your characteristics or your traits from one generation to the other. Speaking of traits, DNA must put genetic work to genetic information to work by determining the different characteristics of a genes, aka traits. So your smile, your mannerism, your skin tone, your hair color, your hair texture, your eye color, all of these are determined by your genetic information. And finally, and what I might also say equally important, DNA must be easily copied because this is what happens every time a cell divides, even for asexual or sexual reproduction. All cell division must have DNA replication if you're going to make an exact copy of the old cell or if you're going to increase the probability of increasing diversity with sexual reproduction. And so your DNA still have to be copied because you can't pass something on if you haven't replicated it, you haven't made more copies of it. All right. So this brings us to the actual components of DNA. Well, DNA is actually made up of nucleotides, and what I mean by that is, nucleotides come in, three parts. All right. First, we have the phosphate group. We have the let's. I'm gonna make a DNA strand over here. So in this, we said DNA is made up of the repetition of nucleotides, so a repeated pattern of nucleotides. So we always start, well, not always, but we have a phosphate group, and that phosphate group is actually, or that phosphate is attached to a deoxyribose sugar, which we call the 5-carbon sugar, which can also be called pentose sugar because pentagon is five-sided geometric figure that we learn in geometry. So we have roughly, don't take my drawing to be too critical, all right? And that 5-carbon sugar in one area is attached to a nitrogen base. So in this one, we're going to pick adenine, or for adenine, all right? And then here you'll have another phosphate connected to a, pentose sugar, deoxyribose. Please always say deoxyribose. It gets you in a good habit. And that's it. This is C, and then another phosphate connected to a deoxyribose sugar connected to it. So it's a repetition of nucleotides, and when I mean nucleotides, is this right here is a nucleotide. This right here is a separate nucleotide, and this right here is a separate nucleotide. So that this one single strand of DNA is made up of 3 nucleotides. All right. So our nitrogen bases come in how many? Again, 4. A, T, C, and G. I kind of like to arrange it this way because then I know A and T bond together and C and G bonds with each other. All right. The nitrogen bases are broken down into 2 broad categories. There's the purines, and the purines have 2 rings, and as far as we go, just know that there are purines and they have 2 rings, and they're made of adenine and guanine. All right, when you hear ring, it's not necessarily going to be a circle, but 2 geometric shapes next to 2 geometrical shapes next to each other. And we have our pyrimidine or pyrimidines, and however you might want to sound, potatoes, tomatoes. Well, same thing here. All right, these guys have only 1 ring, and they're composed of thymine and cytosine. Here's the thing, though, is that when you look at this, to make a nucleotide, to make a base pairs of nitrogen bases, you will need a purine and a pyrimidine to come together because it takes this adenine to bond with this thymine to make a base pair, and it takes this guanine to bond with this cytosine. All right. According to Shawgar's rule, A and T will have a double bond of hydrogen bonds, and so C and G will be triple bond. So sorry, C and G. This is supposed to be a G. There you go. All right. I mean, think about it. The alternating, triple to double bonds creates a flexibility to also have the helical structure in a DNA model. And also, hydrogen bonds being a weak bond also creates add to that flexibility as well. And when you look at it too, because DNA must also be easily copied, you don't want a very strong rigid bond to hold together something that must open up, close up, open up, close up, open up, close up all the time. You want something that's relatively weak but stable. So hydrogen bonds actually, is a better fit for that job. All right, how do we put it all together? As we can see from this image, we have what? Our sugar, phosphate, nitrogen base. This here makes one nucleotide. And every nucleotide has a opposite friend. So we see with the G and C would be our nitrogen bases, they have a triple bond. And the triple bond pretty much has to come in how the orientation of those shape, which sides are, facing each other. And we are not going to go into that detail. Sorry. DNA's backbone is made up of phosphate groups and the deoxyribose sugar. What do I mean by backbone? Look at DNA as a ladder. All right, the ladder has two sides. All right, you have this side and you have that side, and joined in between are the rungs in the ladder. Well, like a backbone would for us vertebrates, your backbone support your structure. So the support structure, and they do more than just that, but for now, are your deoxyribose sugar and your phosphate. So you see phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And the same thing is on the other side. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. It's the same way on the other side as well. Your nitrogen bases would be the rungs in the ladder. All right, T to A, G to C, A to T, and they become the rungs in the ladder. And you have these two strands that are complementary. And sorry, this is complementary with an E actually. Complementary to each other because they complete each other. You need both components to work together because the rung by itself does not hold. The backbones or the side by themselves do not hold. So again, in quick review, in our sides of the DNA molecule, we have the rungs. We have the sides, which are made up of the phosphate and the deoxyribose sugar, and in between are joined together, holding in place by the hydrogen bonds, are your nitrogen bases from both sides, and therefore gluing both strands together in the middle. All right, chromosomes. What do chromosomes have to do with any of this? Well, we first have to understand the different types of cells. Prokaryotic cell we've already reviewed over the prokaryote means before nucleus, so these are cells with no nucleus, and so that DNA is found in the cytoplasm, and it's a single-stranded circular DNA. So for example, this bacteria this bacterium will have is circular DNA, and that's as complicated as that guy goes. All right, so easy. Well, eukaryotic, meaning is after nucleus, or these are nucleated cells. So eukaryotic cells have the DNA inside of the nucleus. All right, pretty much eukaryotic cell, because they're much more complex. They have more than 1,000 times more DNA than a prokaryote. And because, for example, if you look at it this way, you have about roughly 37 trillion cells, in an average adult. Each cell roughly have their own DNA, and the DNA is roughly about a meter long. That's a lot of things to fit in smaller things. I don't know if that makes sense, but that's what. But why why chromosome? Because chromosomes create, such an organizational filing system that that we need to fit all those DNA in the tight little cells that they're from. Look at it this way: when winter is over, You take all your big blanket and your heavy coat and everything else, you put them in these vacuum bag, and you take the vacuum cleaner and suck the air out of it so you can pack a bunch of them in a smaller space. If you were just to take them and just fold them and put them up there, you might even be able to fit 1 or 2 quilts in your closet space. Well, DNA must be folded so tightly in order to fit inside of the cell. And chromosome is the way to go. That's why chromosome. Well, how does this happen? The structure of a chromosome. Chromosome pretty much, like I said earlier, is more than about 1 meter length of DNA, and the DNA gets wrapped around this protein called histone to condense them further. So this is how they make chromatin inside of the DNA, the spaghetti-like looking thing inside of the DNA, as we mentioned several units ago. All right. The way I look at it is kind of imagine a yo-yo. All right. How you coil, you wrap the coil in the middle of the yo-yo, and then, well, but the yo-yo is different, and so the histone will be the yo-yo, and the string, the strings will be the DNA wrapped around, and you do that several times in order to actually create, a very tightly packed DNA in order to be more organized, because this has to be organized. If this is not organized, DNA replication is in big trouble, because um, look at it this way. When you go, I think it's the next, there you go. When you go fishing, I like fishing. If you're going to take your fishing line and spool it around, the, you know, you're going to spool your fishing line. You want your fishing line to actually spool in an organized fashion. So when you actually cast it, that your hook and bit will go flying where you want it to go. But when you do not do that in an organized manner, and then your hook probably sometimes end up slapping you in the face because it doesn't go anywhere or just come back and hit the pole. Well, because for DNA to happen, DNA replication to happen, all of this complex structure right here has to actually, or have to actually open up, and then open up to the strand of the DNA, and then that strand opens up, which we're about to shortly go into, and then start to make copies. So nucleosomes get wrapped around the coil, they get wrapped around to form a coil, and these coils get wrapped around further to form supercoils. Supercoils get wrapped around to form chromosomes. So basically, this is several supercoils wrapped around this right here. These guys are several, supercoiled, wrapped around each other. All right. Basically, this side is, this is the one parent, and this is the other parent's contribution of that same gene. So let's not get confused. All right, DNA replication. Well, we say, this, we said this over and over when we went through mitosis and meiosis. All right, DNA replication happens in the cell cycle. In a cell cycle, we have G0 through G0 through G1. All right, G1, we said the cell is growing. After the cell has grown it to a size, it enters the S phase, and we said S stands for synthesis. And synthesis, what is being synthesized? DNA replication. And then the cell will go into G2 phase, where further growth happen and get ready. And then the cell will go into mitotic phase, which have, prophase, metaphase, anaphase, telophase, and then goes into cytokinesis. And then you have two identical, cells that are genetically the same. Well, how do you create a those genetic identical cells? That happens in the synthesis phase, or S phase. And this is how that happens: DNA replication. Again, why so organized with all of this? In order to replicate DNA, DNA must be organized and easy to unwrap. You can't have something going from this, helical structure, and suddenly, no, you can, you have to be able to unwrap it, because actually, technically, what you're looking at, you have several, coils of all of this going on you know, it's kind of hard to copy that in that form. So DNA has to be able to actually be unraveled in order to be able to make copies of, of of the original strand. So it has to happen, like I said earlier, in a very organized format. All right. Uh, the, there's an enzyme that actually happened before the helicase enzyme. Is that there's an enzyme called the topoisomerase. Topoisomerase will have to come through first and actually straighten out your DNA, so unwraps it, or unwinds it, uncoils it, and then the helicase enzyme will come through and nick right through the hydrogen bonds, holding your nitrogen bases together, and then that will cause your double strand to become you know two single-stranded DNA. Well, and that's just the simplistic summary of a very much more detailed process. Well, first of all, prokaryotes, unzipping occurred at one origin and they proceed in both directions. So, because I mean, think about prokaryotic cell DNA. Again, going back to our simple bacterium cell, it's just a circular DNA. So if you nick this right here, well, you got 2 points of origin of replication. Eukaryotic, although can have hundreds of origins along the DNA molecule. So the more origins you have, the faster that molecule can be replicated. The new DNA strands are built from a free phosphate group, 5' direction to the 3' direction, and we'll talk about more of that soon or shortly. Uh, when I think about the origins, think about a relay. The more people you have in a relay, the quicker you can improve your time to cover the same distance as to one person actually doing it, or even just two people doing that. All right, so here's our DNA molecule, or a double-stranded DNA, as you can see from here, 3' to 5' and 5' to 3'. What do I mean by that? If you look at it, one DNA strand going from a 3' direction, and it'll go up to a 5' direction, and therefore the other side will be running from a 3' to from a 5' to a 3'. All right. So there are several enzymes that come into play when it comes to DNA replication. All right. The DNA strand that goes from a 3' to a 5', we call this the leading strand. All right. It's the leader. Leaders, they stay at the head. You cannot be leading and you're in the back. All right. The other strand going from a 5' to a 3' is called the lagging strand. If you're lagging in a race, you will always be behind. All right. Put that in place. So the topoisomerase job is to come in and straightens out the DNA. All right. Because if you had to cut the DNA half through the hydrogen bond and it was not uncoiled, well, you might be not just cutting hydrogen bonds, because over here you will have these phosphate and deoxyribose that you might actually nick too, and that will create more complication. You will have more fragments of DNA than DNA strands. But shortly after the topoisomerase come through and uncoils or unwinds the DNA, is followed by the helicase enzyme. And what the helicase is like the molecular scissors that come through and nick the hydrogen bonds, aka why we want the hydrogen bond to be a weak bond, because it's easy to be nicked. And so now your DNA coils and opens up. Well, you have these single strand binding proteins that sort of act like, to hold the templates in place so they don't, stick to each other. Think about when you go, and you ladies might be more to, more, you might know more about this, but when you go to the spa, thank you, I kind of blanked out on that, and you were to do a pedicure and or manicure, and your nails got done, they stick this little finger-like thing in between your toe or your fingers, so that your nails don't touch each other until it dries out. Well, something similar. So these guys will hold the templates into place until after the new strand has been made. So topoisomerase come through, uncoils, helicase comes through, and opens up the hydrogen bonds. Well, make a guess. Which strand do you think will copy quickly first? Well, if you say the leading strand, ta-da! Because here's the thing is that the leading strand will copy quicker because the leading strand starts out with a 3' to 5' direction. All right. So the new strand, aka the complementary strand, would need to run from a, you know, 5' direction to a 3' direction, which is what we saw up here. All right. And hey, guess what? Who makes the new DNA strand? It's called DNA polymerase. All right, there are different types of DNA polymerase. For as far as we go right now, let's just say DNA polymerase. But DNA polymerase is programmed. He prefers to go in a 5' to a 3' direction. And guess what direction is this strand going? 5' to a 3' direction. So copying that new strand or complementary strand with an E, that is an easy process for the leading strand. It's not complicated. It just zip, 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 and go. All right. Well, the same is not so true for the other strand or the lagging strand because the lagging strand is going from a 5' to a 3'. All right. So its complementary strand is going to go from a what? 3' to a 5'. That's not the direction of the polymerase. So therefore, because it's not going in the same direction as a polymerase, like I'm going to one city and you're going to the other city, and we're going in opposite direction, then you're asking me for a ride. That doesn't really work very well. So certain things have to happen first. So what has to happen first is that we have to lay uh, some building blocks along the way, and these building blocks are called RNA primer. And you say, wait, we're talking about DNA. Yes, RNA primer. RNA primer run in a 3' to a 5' direction. So that kind of lays the foundation to have the polymerase set up to go from a 3' or 5' to a 3' direction. So then the polymerase comes in and add in the DNA sequences. But this polymerase is lagging behind because all of these things have to happen first. And you also have this other enzyme called the ligase, which comes in to close these gaps that we have in between the primers. All right. Well, because those gaps or these gaps are called Okazaki fragments. Because remember the primer is just little bridges being put in place to prime the polymerase what to do. Just like if you're going to put on your makeup, ladies, you need to put on a primer if you want your makeup to stick. Or like you guys who like, well, guys, if you like makeup too, that's up to you. But same idea with painting a wall. You want your paint to stay and not peel off or easily be removed. You gotta, and that's the first time you're painting that structure. You have to use the primer. So just in review, for the lagging strand to be able to be copied, you need a primer to come in and lay the bricks or the foundation or the priming factor, which are RNA sequences, and then followed by then the DNA polymerase can come in and add the DNA sequences, but not completely done because you have these Okazaki fragments, but those Okazaki fragments are closed by the DNA ligase, which add in phosphate to close those gaps, because the phosphate is also one of the backbones of the DNA, which will be the sides. All right. Once that is done, all right, you have then a nuclease, another enzyme will come through and proofread. I know this picture is getting messy. Proofread all both of your new strands. And once it does that, if there's any mistake, it will remove those mistakes, those bad nucleotides. And then remember, the polymerase always add in a new nucleotide. So after the nucleus remove the bad nucleotides, then the polymerase come back through and then add in the new correct nucleotides. And then your DNA, as far as this class go, is being made or has been made. So then you have two identical DNA strands because they come from the same source. Well, having that said, DNA replication is called a semi-conservative process because see the strand number 1 here and strand number 2, well, guess what? They are based off of a template that comes from the original DNA. All right, so those strands oh, sorry, the original strands can also be called the templates. Some people call it the parents, strands, which will give rise to the complementary strands, all right, or the daughter strands. Now that we just went through a very messy picture. Let me see if we can clean things up a little bit here. So you start out with, oops, bad one. Where's my eraser? There you go. All right, so we start out with DNA strand, helical. We know you can't copy the DNA in that. So the DNA actually get, straightened out using a topoisomerase. And then the topoisomerase, so now we're going to have our DNA open up by the topoisomerase. But here's what's going to happen is then, here, this is called a replication fork in between here, this space. This is called a replication fork, all right, because it kind of looks like a fork, all right. What's going to happen is then you have, if this is your, 3' to 5' direction and 5' to 3' direction, all right. 3' to 5', this will be our leading strand. So this strand will copy first because the DNA polymerase runs in the other direction, 5' to 3'. So it will start copying in that direction. All right, so that's quick. Well, this will become our lagging strand. All right, and you will have, its complementary strand will want to be going from a 3' to 5' direction because that does not happen. So the polymerase which actually add in this complementary strand or the new nucleotide cannot go in a 3' to 5' direction. So it's actually going to be preferring to be going in this direction. So the lagging strand actually copy in the opposite direction as, with the leading strand. So why does it do that? Because you need to add primers in place to lay the foundation for 5' to 3' direction. And those primers then leave the Okazaki fragments or gaps in between. And those gaps are closed by the DNA ligase, which add in phosphate. And then your polymerase comes through and completes the whole setup. Each of these strands are actually edited by the DNA nucleus that comes through and proofread them. If there's any bad bases or nucleotide, it removes them, and then the polymerase comes back in and finish the job by adding in the correct base pairs. And then you have two identical DNA that can go into two identical new cells or daughter cells. All right, and this is just a very detailed description of what we just went through with pictures. So if you are hung up over those pictures and it's too overlapping, here you go. Use this to break things down for your come back and forth and to kind of pace yourself, because I've described these in a very sequential order with pictures on the side. All right. Yeah, so this concludes our podcast on DNA replication. I hope you found this helpful for you, and if you have questions while I'm gone, please consult this to clarify things. And whatever you can find here, always know problem solving is always our best solution. Go to Professor Google and look things up, because we are problem solvers.